structure of the DNA molecule was discovered in the early years of 1950s. Note that the structure was discovered, not the molecule. The discovery of DNA molecules predates to the 18th century. But DNA was ignored for a long time, and it was proteins that grabbed a lot of attention. However, post-Griffith's transformation experiment, the other two notable works in the timeline helped shift the focus towards DNA. So around the early years of 1950s, an English chemist named Rosalind Franklin started working on DNA molecules. She isolated the DNA crystals and shone X-rays on them. As a result, she obtained fine images of the structure of the DNA molecules. According to sources, the best one looked somewhat like this. Around the same time, Francis Crick and James Watson, two scientists in the field of genetics, worked on the structure of DNA and reached a similar conclusion like Franklin did. However, according to sources, they were fortunate to receive better and earlier recognition compared to Franklin. They even shared a Nobel Prize with Maurice Wilkins for their discovery of the double helical structure of DNA. So let's understand how exactly this double helical structure looks. We may have come across the structure of DNA somewhere. Is this how it really looks? Yes. According to Franklin, Watson, and Crick, the DNA is made up of three major components. One is a pentose sugar molecule called deoxyribose. How is this obtained? It's obtained by removing one oxygen atom from a ribose molecule. Thus the name deoxyribonucleic acid. The second component is phosphate group. This helps bind the two deoxyribose molecules together. The third and the fifth carbon atoms of the adjacent deoxyribose molecules are bound by the phosphate groups. Lastly, the nitrogenous base forms the core of the double helix. These are four different molecules that bind in pairs. Two of them are called purines, and the other two are called pyrimidines. Adenine and guanine are the two purines which bond with the pyrimidines thymine and cytosine. Can any purine bind with any pyrimidine? Nope. It's a thumb rule that adenine always binds with thymine, while guanine binds with cytosine only. These molecules are bound to each other with the help of hydrogen bonds. Adenine binds with thymine with the help of two hydrogen bonds. Guanine and cytosine, however, bind with three hydrogen bonds. Did you know that one unit consisting of deoxyribose molecule, one nitrogenous base, and a phosphate group is called a nucleotide? And what if the phosphate group is removed from this nucleotide unit? It's called a nucleoside then. Nora wants to know if there's any way of measuring a DNA molecule. What about its dimensions? Watch the next part to know more. The dimensions of DNA are very simple and easy to remember. The diameter of the double helix is around 2 nanometers. The double helical strand has one major and one minor groove that makes up one turn. The length of one turn is around 3.4 nanometers. And there are around 10 base pairs in one turn. Now, can you help me with the distance between the two adjacent base pairs? It's simple. The distance is 0.34 nanometers. It's simply 3.4 divided by 10. This is how the molecules are arranged. So in this double helix, this portion is the backbone, which is made up of the deoxyribose molecules and the phosphate groups. Whereas this portion that forms the core is made up of the nitrogenous bases.
This is structure of DNA, double helical structure. Just now you have seen the video. In that, what are the components present? Deoxyribose sugar, phosphate, nitrogen bases. The nitrogen bases are two types, purines Wait, let me change the color. Purines. These purines are two types. Guanine represented as G, adenine represented as A. Next, pyrimidines. Pyrimidines are two types, cytosine and thymine. Cytosine is represented as C and thymine is represented as T. Now here there is a combination that one purine will always unite or fuse with one pyrimidine, but no two pyrimidines will fuse. But there is one more combination that always adenine will fuse with the thymine and cytosine will fuse with the guanine. So there is a point that in the previous video, today's video, you have seen that these in between these two hydrogen bonds will be present. Between adenine and thymine, two hydrogen bonds will be present, and cytosine and thymine there are three hydrogen bonds. So when bonds are not matching, it cannot be fused or bond cannot be formed. You learnt in the chemistry. So always the combination is like that. Now come to the DNA structure. Here, this is the DNA. Now the DNA is in this way. Adenine A, T, C, G, G, C, like that. I think you are able to understand. Like how we open zip, like that, it will get opened because we are talking about, now our topic is reproduction. So in this reproduction, cellular level the reproduction should take place, and this is the DNA molecule. The DNA is nothing but the chromosome. So chromosome first should divide, then only the equal number of chromosomes will be distributed to the cells. So now how the DNA actually replicating or duplicating, I am going to explain. Now here you see, like half piece, it will become half piece, and when it becomes half, now you see this T, A are got separated. Now T will get another A. Let me represent with another color. So this will get A, this C, G will get C, C, G, T, A, T, G, C, in this way. Now this side. Let us go to this side. As there is no place, I am writing here. Okay, I will write here only so that you will understand. T, G, C, T, A, G, G, C, C. Like that, how many DNA molecules are formed? 2. So, replication is over. This will push it to one cell. This will push it to another cell. So, cellular reproduction is over. Cell is divided. Once cell is divided, now the organism is ready to divide. Not only DNA. DNA is the information source. That what are the qualities, what are the behavior habits, or whatever it may be. Everything will be represented in this DNA. Even we are understanding that, we are reading in the newspaper that corona is changing its DNA so frequently. That's why it is becoming, making the vaccination is becoming a challenge. So we are reading the news because we are in that COVID pandemic. So this is the nature. Actually our genes will not change, our DNA will not change so frequently, but the DNA of virus is having that tendency to frequently changes its DNA. Now, you see, one more important point I wanted to share you, that is variation. What is it? Variation. What do you mean by variation? Small differences are called variations. How actually small differences will occur? Now you see, when this duplication is taking place, it will done very fast. When the teacher has written 2 pages of notes on the board and ask you to copy very fast, so you may, there are a lot of changes that you may go so many mistakes. Now here when the DNA is duplicating, there may be some mistakes. So what do you call that? You can say errors. What you can say? Errors. Errors in DNA copying. So because of this errors in DNA copying, what happens? Small variations will occur. Very small variations. So these small variations, and sometimes it may be useful, sometimes it may be harmful. If you have that very useful variation, then you can survive better in the society. Okay? Followed children. Now one paragraph is there, how bacteria is able to survive in changing situations and changing environmental factors, that we will discuss in the virtual class. Right? Now variation, did you understand children? Why actually variations occur? The small inaccuracies in DNA copying or The errors that occur in the DNA copying makes the individual to get variation. Right?
Variation. Some variations take place each time a DNA is copied. So, the DNA copies are similar but not identical. Hence, the offsprings, though similar to parents, are not identical. These variations play an important role in the survival of a species in unfavorable conditions and prevent wiping out of a species due to drastic changes in the ecological system. For example, varying temperature and varying water level.